Hey, everybody. So we're just going to jump right into chapter 24 and get through 24 and 25 here in one big swing so we can finish up unit 2. What we're going to be talking about in chapter 24 to start with is microbial ecology. And that's going to really just talk about how microbes live and where they live and everything like that. They're like, biomes, which we'll talk about that term. And we'll talk about roles of producers and consumers and decomposers. So that's having to do with like the food chain, essentially. And then carbon, nitrogen, hydrologic, which is the water cycles, and how organisms live in them, how they're affected by them, and what role they play in the entire cycle overall, which is actually pretty significant, especially within, the organic environment. So microbial ecology is the term referring to the study of microbes in their natural habitats where they're normally found, not like necessarily outside of their normal areas. So ecological studies are going to look at the biotic versus the abiotic factors. Biotic is the living stuff that might affect the microbes and how they live and where they live, and the abiotic factors are the non-living. What will we care about the non-living? Those are actually pretty significant. We're talking about atmospheric gases, minerals, water, temperature, light, pH. Yeah, so things that live in acid pools, that's an abiotic factor. Things that live in, hydrothermal vents, that temperature is a abiotic factor. So the biosphere is a term that refers to all physical locations on Earth that will support life. We're talking about dividing it into the terrestrial versus the aquatic realms. Okay, so that's one way we can divide it: terrestrial versus aquatic. Terrestrial, obviously land; aquatic, obviously, freshwater and marine realms. You can divide that even further. So then you can also divide the biosphere in a different way, which is the hydrosphere, which is anything with the water; the lithosphere, which is like basically the land portion, just for basic argument, okay? And then the atmosphere, anything up into the air. So those are the 3 layers that we're talking about. And then within that, we have the terrestrial versus the aquatic realm, portions of the biosphere where, life can be supported. So a biome that's a term that refers to the climatic regions, each of which are characterized by dominant plants, temperature, and precipitation. What does that mean exactly? Well, we're talking about things like the rainforest. We're talking about things like the tundra. That's a biome, right? So whenever you learn this stuff in grade school, that's what we're talking about, okay? Biome, climatic regions. These, temperate forests versus, the desert, how much rain they receive and what sorts of organisms grow there, what sorts of plants, what sorts of animals are supported, all of that, okay? Those are the biomes. We have communities. In these areas, we have communities and populations. A community is a clustered associated association of mixed assemblages of organisms that live together in the same place at the same time. So we're talking about everything that lives in that locale, not just one species, right? Not just one kind of organism. Whereas a population is a group of organisms of the same kind. Okay, so we have a group of, the turtles that are all the same, or the human beings. That would be a good example, right? Because we know we're all the same species, and stuff like that. Coyotes, whatever. But community, if we're talking about coyotes, if we were talking about coyotes in like the desert, we might say the coyotes and the rabbits and the scorpions and the microbes there and whatever else lives in the desert with that, right? That's the community. That's the difference. Okay. A habitat, so that's a term used to describe physical location environment where an organism is adapted to live. The microenvironment is the particular qualities of that area that is suitable to that organism. Okay, that microbe, like E. coli, like whatever it specifically prefers to grow, those exact qualities. That was including the gas, like oxygen, light, temperature, all that stuff that makes it happy. And a niche is an overall role that the species plays in the community. So everything has a role. It has some sort of way that it works in with all the other organisms that's being allowed to subside there. Because there has stuff to provide energy, and it's probably providing energy to something else. How is that working out in that system? That's the niche, its role in that community. So that has to do with its nutritional intake, okay? And then the position in the community structure, what is eating it, what it is eating, that sort of a thing. So talking about that food chain, right? So the thing at the bottom, which is eaten by the thing above it, which is eaten by the thing above it, and so on and so forth. And then the rate of population growth, how fast and quickly it will grow in comparison to everything else. So this is how energy flows within these systems. It always starts with an energy source, which typically, we're going to talk about there are different ones. It doesn't have to only be light, but typically we're talking about light, like photosynthesis, photoautotrophs, okay? So we're going to break these terms down, but autotrophs, they're going to be making their own energies. And then we have, as far as like their energy sources are concerned. And then we have things like, the heterotrophs that have to consume something. So anything that says consumer over here is a heterotroph, okay? And we also consider decomposers to be heterotrophs because they're breaking down a whole bunch of other stuff that came from other organisms that built those things, okay? The primary producers down here, these are the ones that make their own. So they make their own food, their own carbon source, they make their own energy source, whatever, but they get it all for themselves, right? And everything else is going to depend upon it to sustain its life, right? Like plants is what I'm talking about here primarily, yes. Primarily plants, there's bacteria that fall under these as well, cyanobacteria and stuff like that are going to make their own energy and have their own carbon source, but you get it. Okay, primary producers, we have photosynthesis, algae, bacteria, sulfur bacteria. So these are microorganisms. Plants are not microorganisms, yeah. Chemosynthesis, that means they're getting the energy essentially from chemicals, not talking about light anymore. So usually chemicals and the thermal vents. And using a different system to get that. Then we have the decomposers. Decomposers are just going to break down into dead things. And then we have cycling agents for biogeochemical cycles, talking about recycling of carbon, nitrogen, phosphorus, sulfur. If you guys aren't aware of this, it's extremely important in agriculture right now, of nitrogen, there being not enough nitrogen in the soil. That comes from certain bacteria that are usually incorporated by the root systems of plants, and they're just not getting what they need from the soil because of change in the climate, essentially. And then we have parasites. Parasite is a term of anything that lives and feeds on a host. It has to do that. Doesn't have a choice. So if we were talking about single-celled algae like Chlamydomonas, I forgot there are questions in these. If we're talking about single-celled organisms like algae, and an example here they're giving us is Chlamydomonas, likely to perform in an ecosystem. So algae, we were talking about how they were photosynthetic, right? Photoautotrophs. So if they are a photoautotroph, they're going to be a primary producer because they produce their own. And the primary because they start from scratch. Okay, primary producer. What are biogeochemical cycles? This is literally the recycling of the elements in the living systems. All elements ultimately originate from non-living, long-term reservoir in the atmosphere, lithosphere, or the hydrosphere. Elements cycle between abiotic environment and the biotic environment, of course. And then we know it has to come from somewhere, right? Didn't start within living things. And then recycling maintains a necessary balance of nutrients in the biosphere so that they do not build up or become unavailable. Right. So if the system ends up being shifted somehow, that's how we could lose them from one area into another or whatever. So moving on. So these cycles are complex systems that rely on interplay of primary producers, right, creating energy, the consumers that consume and upwards, and then decomposers that break down the dead things. All organisms participate in the recycling. All of them do. But only certain categories of microorganisms have metabolic pathways for converting the inorganic compounds from one nutritional form to another so they can be used by something else. Okay, that's where we get into talking about like the nitrogen. I'm going to start with the carbon cycle though. You guys know about breathing because we've talked about it already, but we start off, where do we want to start on it? We have free carbon dioxide in our atmosphere, whatever. The trees might use it and incorporate it into its sugars, into its cellulose, and whatever else, right? To form its own cells. So it's using that carbon from the atmosphere, carbon dioxide, okay? Then this can go on to be, eaten up by consumers, like animals and protozoa and all sorts of, I don't know why it has to only be micro in this picture, but it's only micro in the picture. But essentially, yeah, so the free CO2 in that tree gets incorporated into that, and then organisms eat it, essentially. And then that gets incorporated into those organisms. And then that can be decomposed when they die by fungi and other bacteria that we call saprobes. It's the term for them. You'll learn it. And then we have, that leads to respiration as well. We have release of CO2. Sorry. I don't know how to even. Cool. Or I had a pop-up thingy pop up because I clicked a button accidentally. So respiration is what we're talking about with release of CO2. Now, so we will release, this is animals, by the way, down here. This is where we would be considered to be part of. And the microbes as well. Going to respiration to give off carbon dioxide back into the atmosphere, which can be used back up by phototrophs or autotrophs in general, we should say. Okay. Moving on, the carbon cycle. Basically, this is just showing about how carbon dioxide measurements over all this time for over 2,000 years. This is over 2,000 years. And then people came along and started the Industrial Revolution. And this is the change in the carbon dioxide as of that, after that started happening. You can't say that people didn't have an effect on that. So it's idea there. And then talking about global warming a little bit more, if you watch this figure here, it's indisputable when it gets to it. We have in the 30s and 40s, and then as we have grown industrially, you will see the global temperature change. Right? It's moved outwards. It's not, you can't argue it, right? It's like I'm just saying. So the color has to do with the temperature. So as it veers more towards red, that's higher temperature. This is every year. After that's where we will watch it one more time. This is real. It's actual data. So there you go. I mean, at this point, it's indisputable. It's not even a question, that part of everything being, as far as global warming, as far as climate change goes, that's a real thing. So the question about that becomes, what do we do about it? Is there anything that can be done? And I think the consensus, I think that people have gotten to, if you correct me if I'm wrong, I may be wrong about this. I think the consensus was that we can't do anything about it at this point. Like we've reached the limit of time that we have to deal with it, essentially. I might be wrong about it, but I thought that I had heard something about that. Anyways, either way, it doesn't matter, right? We should still try to make the world a better place for the people coming after us. No matter what. So that's all that this slide is really getting onto us. What if it's a big hoax and we create a better world for nothing, essentially? Even if you don't agree with global warming or climate change, and that's fine. I can't make you do things. So believe what you want to believe, but we should still make the world better. Don't even try to argue with that one. We should still work towards making it better. I don't know how necessarily, so don't look at me for that, please. Okay, nitrogen, moving on, nitrogen cycle. This has to deal with the global warming effects as well. In a minute, we'll get into that, but we have the 4 basic kinds of reactions that occur when we're talking about with nitrogen. We're talking about nitrogen fixation, ammonification, nitrification, and denitrification. These are pretty important terms that you might want to be familiar with, kind of know these 4 terms. Okay, I'm not going to make you know what each one of them specifically does, but be familiar of the 4 basic types of reactions. If I were to say, what are the 4 basic types of reactions that are important in the nitrogen cycle, then you might want to be able to list these 4 off the top of your head. Okay, that's the idea there. Nitrogen cycle, we have a symbiotic relationship between rhizobia and legumes. Legumes being like beans, right? Rhizobia being bacteria, and these bacteria will they're gram-negative, motile, rod-shaped bacteria. I don't know why you need to know that, but there is the information about it. Right, so the legumes have little, they form these root nodules when they get infected with the rhizobia. They become symbiotic because the bacteria will convert the nitrogen in the soil to a usable form for the plant. So the bacteria are getting to, be secured within these root nodules and be kept safe, and get pushed out into the soil where that's all nice and whatever, and there's nutrients for them in there, whereas the plant is benefiting because it's getting the byproducts of the bacterium. So that's and that's a system, basically, essentially the enzyme system of rhizobia supplies a constant source of reduced nitrogen to those plants, legumes. This is an example over time when we look at a field that was growing like this in the past, and now what fields are looking at now. This is actually a real thing as well. It's a very serious issue at the time. Moving on, other elements. It's not just about carbon and nitrogen, or even oxygen, but there's sulfur, there's phosphorus, and there's metals, right? So sulfur is important in the environment. We need it for like cysteine. Well, it's an amino acid. Okay, so we need it for amino acids, certain ones. It's very important because it creates certain bonds in the proteins that are important for the shape of the proteins. Phosphorus, we need phosphorus to make the phosphate groups, like we use ATP, adenosine triphosphate. You need phosphorus for that. Yeah, that's the P in it. And then we have metals like iron. You need iron in your body, right? So like that's what's in your blood that helps you carry oxygen from place to place, right? So there's other things that we would need or that could become involved in our bodies, or even the microbes. There are even microbes that can metabolize uranium. So that's pretty cool. I don't know that they should exist, but they do. Okay, so this is just showing kind of that life cycle that's going on in the soil microbiology. And we don't need to go through all the little details, but you can see the little, bacteria hyphae that are growing off on that little root piece, and then how they're interacting with its the environment around it. There's a mite, there's all the nematode, worm, and all these little creatures that can live in the soil and depend on one another for nutrients. It's like their little, community, like we were talking about earlier, right? That's their community. So deep subsurface microbiology, we're talking about here digging way, way deep down into the ocean, sampling in 2 miles or more deeper below the surface. We found bacteria 30 meters below the seafloor in clay, below the seafloor. So getting down under the sea, then below the seafloor, another 30 meters into the clay there. That clay hasn't been disturbed for 86 million years. Nothing has been down there for 86 million years. And we have discovered microbes that can live there. We think they might have some secrets to, life and how to longevity and stuff like that. Really, honestly, it's just because they grow super slow. It's not because of anything crazy. Yeah, so water, you guys have seen these cycles before, surely. The rain makes the precipitation that runs off, and it runs into the ocean, and the ocean into the groundwater, or into the streams and the rivers, and then all of this stuff interacting with one another. Yeah, so that's about water and how it gets cycled just generally in the environment itself, not necessarily within animals or creatures or anything. So about in our fresh water, fresh surface water, lakes comprise 87% of that, rivers 2%, and swamps 11%. And fresh water outside of that, that's retained outside of just fresh surface water, this liquid. So we're talking about here ice caps, glaciers, groundwater, things that we can't get to on the surface there. Okay. When you break it down, this is 0.3% of all the fresh water is the fresh surface water. So this is how it's breaking it down. I should have started at the bottom. I started at the top because, you know, I just saw top and I thought maybe we'll start here. Sorry. So we start down here. We have all the water, saline oceans, 97% of it. And then 3% is fresh altogether. Then 0.3% of that entire fresh, 0.3% of it is the fresh surface water, the lakes, the rivers, and swamps. Okay. So trying to break it down, man. I should have started at the bottom. My bad. My marine environment's going on. The salty, the salinity, it's not all the same. Like we would think of like water, you know, going into, you know, a lake or whatever, and then going into an ocean, and you're like, oh, this is fresh water, this is salt water. But is all the salt water exactly the same? Certainly not, right? So you have mixtures at points where it's going to have variations in the salinity, how much salt there is in it. Variations in the depth, obviously. The pressure, so if you're going super deep, there's more pressure there. And mixing, so how the different levels might be mixing, because you know lakes don't mix as much as an ocean. Yeah, so that's the idea with that. There's this guy that did deep sea sampling, and you know, he found out that essentially when he was sampling all throughout the oceans, there were 10 million viruses per milliliter of sample that he got from the ocean water. Most of those are bacteriophages. So, you know, bacteriophages are viruses that infect bacteria. All right, this I know, we can watch this, I suppose. I don't think I'm going to play this. If you guys are interested in Googling this, it's a pretty interesting little TED talk about what I was just saying, essentially. This one studies deep sea microbes, and they pulled out the deep sea sample, and those microbes were still alive. Well, how could they possibly be alive? And can that lead us to understanding, you know, the longevity of life? And it turned out essentially that those microbes grew, they couldn't grow them in the lab at all, no matter what they did. They gave them the food that they wanted, but they just weren't growing, and they couldn't figure out why. It's just because the cells didn't divide. They didn't need as much energy. So like if she has a pretty good explanation of it, but like if you take a pineapple and hold it at her waist height, she was saying, just because I don't want to stand up, I'm sorry, and then drop it, it was something like, you know, 1,000,000 times a day. I wasn't 1,000,000, but you know, 3,000 times a day, then you generate enough energy within that happening that could power a whole human for a day. But like these bacteria had like, if you dropped like, what did you say, a grain of salt from like 1/10 of a grain of salt from like the height of, you know, her head, and then let that fall down, then that would supply that organism for its entire lifetime or something like that. That's how much energy it needed to live. So it's not the exact, but it's the idea that matters. If you want to watch it, feel free to Google it and watch it. It always, when I play this video, it always deletes my videos off of YouTube. So because it's copyrighted. So I'm sorry, but I'm not playing it right now. Maybe another day. So that's it for chapter 24. Now don't get too excited because we still have to do chapter 25. Okay. So chapter 24, we talked about the ecosystems, the cycling of the elements, and the water cycle. So here you go. This is one from Dr. B that I left in there because it is cute. And what kind of an animal is this? Of course, you guys aren't getting any points for it. So this is a Tasmanian devil. They make some crazy noises. If you guys don't know, Dr. B used to run the micro program. I took it over. He's now the division head for STEM, and he's from Australia. So he likes to put little Australian stuff into his slides, and he, you know, I was bequeathed to them or whatever. And I've edited them down, but to my liking, but some of his stuff is, I left it in there because it's cute. So that's the point. Chapter 25, we're going to talk about drinking water, wastewater cycling, and how we are going to clean sewage, differentiating between the processes for beer and wine. There used to be way more slides about the beer in here, and I took it out because I don't understand it. Okay, you're welcome. Cheeses and how cheese has different flavors, factors involved in safely producing food, all of that. So in the industry overall. So biotechnology, How do we use it within our daily lives? Fermentation of wine, obviously making wine. We also can get vinegar, that pathway as well. If you've ever heard of red wine vinegar. Genetic engineering methods are used in technology as well to make microbes do what we need them to do. They have applications in industry, medicine, agriculture, food sciences, and environmental protection. We also have, like, a lot of times whenever you get your food tested by certain companies, which I used to do this for a living, which is why I know. If you send your company, like McDonald's, which I tested their food, we get sent off to a company to get tested to see if it has any E. coli in any of their burger patties. And they have like a shipment that they're waiting in like their freezer that they don't load into their trucks and send out until they get the test back. And it takes a while. It takes about 12 hours to do the entire test. But aside from that, like growing it up and everything like that, and then running it on a PCR machine, right? So you guys know what PCR is now. Anyways, my point is we had to run it against a standard so that we knew that the test was running. So why do we do stuff like that? We will run a positive that we know is E. coli. Then we'll run a blank too right next to it, and then run all the samples for the test, or we'll put them at the end. It doesn't really matter, but we'll run them with it at the same time so that we can say this run ran properly because our positive was positive and our negative was negative. So if all of these samples came back negative, we can trust that result. So that's the idea with that. So that's where I'm saying we have developed E. coli strains for use for that purpose, that we use in stuff that you wouldn't even really think about that we use microbes for. We need microbes to test for microbes. Fermentation is a term talking about, we're talking about breaking down a food into acids and gases, basically, essentially, is what it's going to be. So what do we use fermentation for? Culturing microbes allows us to create certain organic compounds like acids, like gases, and whatever, like we're putting gas into like cheese, you know, or like the bubbles in your beer, all of that, that's fermentation. So, and we can also use microbes like this to help break down sewage. Why is this not? And break down other bad things in our environments. We can manufacture bacteria to do these specific things for us. Weird, it felt like the screen was moving. I don't know, probably just I'm on steroids. I had pneumonia, so probably all it is. Drinking water, yes. So our drinking water, most of it comes from rivers, aquifers, and springs. We usually don't have water in its natural form, right? It's usually treated somehow. If you have well water, it still has to in some way go through some sort of treatment to be considered safe. It has to be tested at some point, right? So that's the idea there. So even that water had to be tested at some point to confirm that it was safe. We test to be sure that you can drink it. That's that term of being something is safe to drink. It has to be free of pathogens. Pathogens are things that make you sick, right? Microbes that make you sick. That term is potable or potable water. Do you need to know that term for a test? Potable water. Okay, I can barely see this and I apologize for that. Let me see, I can see it over here. We have, yeah, because I like this. I like this slide. Okay, we're going to start here. First, we have water catch in a reservoir, and we'll go through like a big screen. It catches all the big stuff, right? So this screen down here is going to catch like rocks and big stuff, right? Then that's going to include, you know, household waste and stuff like whatever, untreated water. Okay, so number 2 is this one. Then we're pumping it into a holding tank where aeration takes place. Why would we want to aerate it? Why would we want to put air into it? Why would you want to do that? Mix it with air. What's the point in that? Well, you guys haven't really gotten there yet. But some microbes that are anaerobic, we had that term before, they can't do air. They don't use oxygen. Those microbes that are anaerobic, which often grow in sewage situations, they're killed by oxygen. So we try to aerate it to get that going. So starting killing microbes already by aerating, just by putting air through it. All right, then the next part, we are number 3, further settling, separating everything out at this point, and then filtrating through coal. I say coal, but you guys know, understand this, like charcoal. And then sand as well. So we're filtering it even more, settling, filtering, and then we have the addition of chemicals like chlorine added in to kill everything. So we've had chlorine gas treated water, the bubbling chlorine through it. Chlorine gas will kill you and I as well, to kill everything else that's left in there, if there is anything. And then it can go into a storage tank and it can be stored for, later usage. Then you need to drink something in your house, and the treated water will get into your house. Why can't I write this over here? And then you drink it and whatever, and then you pee it out, and then that's where your household wastewater will go back through. So that's the water. That's how sewage is dealt with. Yes. All right. Wastewater treatment, drinking water. So like we said, source comes from rivers, lakes, or wells. We want to get the big sedimentation out. We'll filter through alum. Doesn't matter what it is. Big filtration, little filtration, and settling. And then use antimicrobials like ozone and chlorine. Usually a gas to bubble through it to kill everything else off, and then we can store it for use later. We want to monitor for these pathogens. This isn't everything, but these are prominent ones that we want to test for. You want to be sure that you have them within acceptable limits. So some of these you can have them. They just can't, you don't want too much of them. Yeah, that's real. So Giardia, Cryptosporidium, Campylobacter. If you guys haven't heard of a lot of these, then you probably, I don't know, live under a rock or something. But yeah, Giardia is pretty bad. Cryptosporidium. Campylobacter is the #1 cause of bacterial diarrhea. And we have Salmonella and Shigella, Vibrio, which is like cholera. Mycobacterium, that's like tuberculosis. And then we have hepatitis, Hep A, that's another one. It can be just in your environment. And then Norwalk virus. So, bad stuff in your water. There's a lot of assays that are focusing on water purity to detect fecal contamination. And how would they do that? We have indicator bacteria that are species that are intestinal residents of birds and mammals that can be readily identified using routine tests, that if they are present, so we can put that bacteria in there, set it through the treatment process, and if it comes back out, we aren't doing it right. So they should be killed off. So the other term that you want to know is indicator bacteria that we were just talking about. We use it to make sure the tests are working. Then we have coliform bacteria. These are things that are essentially going to be found in feces, fecal contamination. So that includes E. coli, Citrobacter, Klebsiella, and Enterobacter. Most of these are going to be gram-negative. They are going to ferment lactose, and they're going to produce gas. All right, water safety and our indicators. So we talked about the indicator strains and adding it in, right? You can also look at coliforms as an indicator of those other pathogens. As far as like, if we see E. coli in there, then probably the other worse ones might be there as well. They're not being killed off. So we can use those as indicators themselves. And so anyways, whatever, we get our water sample, we put it into a filter, it filters out down into the bottom, this super fine filter. We take that little filter off, it's got little hash marks on it so we can count better. We put it onto a auger plate or something similar, and then count the colonies after we let it grow for a little while. And that will give us an idea of how many coliforms, so that's poop bacteria, are growing in it. And is that within an acceptable range or not? Is it healthy enough for people to consume? So these are the ranges. Drinking water can have we have to have less than 1 colony forming unit per 100 milliliters. So when you take a 100 ml sample of water from drinking, it can only have less than 1. It cannot even have 1 colony forming unit. You have to go up to like 1,000 in order to even see 1. So recreational water, we have less than 180 colony forming units. And then treated wastewater that's getting pumped back out into like the river systems and stuff. That's less than 400. And yeah, less than 400. So that can have a little more. Wastewater sewage, right? So we like to treat our sewage before we put it back into the environment, right? That's the good idea. We can remove large particles, nutrient levels. Sorry, we can remove large particles. We want to be sure we control the nutrient levels in whatever sample we're adding to whatever, so that it doesn't, isn't friendly to bacteria, right? And then we want to be sure it's free of pathogens before we put it in the environment as well. That's a biggie. Like we were just talking about with those colony forming units, the 400. Primary treatment includes removing the large matter, the solid pieces. The secondary treatment is biodegradation, which is the biodegradation of creating sludge. Bacteria can actually be added into this to break it down even more, the particulate that's in there. And then that sludge can be retreated or whatever, aerated, whatever. You can retreat it a few times and before you have your waste, but your solids that we can get rid of. Then we have the tertiary treatment where we have chlorination and UV treatment to kill everything off. And then that leads to reclaimed water that is safer for people to be in and not quite as dangerous as poop water. All right, this is the idea of what we just talked about. We have our solid wastes. Then we have the secondary stage. We have a sludge digester, trying to break everything down and getting the liquids separated from the solids. We get rid of the solids. We're going to dispose those, or we can reclaim them with an anaerobic digester. So anaerobic bacteria will break down those sludges even further. Then we have, the water that's left on top that we can filter out. And then we want to remove any of the solids that's involved from filtering. And then after it's been chlorinated, we can actually release it into a body of water, as long as it's less than 400 colony forming units, right? That's how it's done. Microorganisms in food, we have the beneficial microbes, right. Food is fermented or otherwise chemically changed by the addition of microbes or microbial products to alter or improve flavor, taste, and texture. That's a beneficial thing in our food. Sorry, I have like sticky lip gloss on. It's really annoying me. The detrimental side of it, bad things associated with microbes and food. They can cause foodborne illness and they can cause food spoilage, which can in turn lead to foodborne illness. Making bread. So right, yeast, yes. Because when the yeast, the Saccharomyces cerevisiae, that's yeast that you are adding in order to, get your dough to rise. That's fermentation that's going on. So the yeast produces volatile organic acids and alcohols that impart certain flavors or aromas. You can also get affected by atmospheric bacteria, like Lactobacillus San Francisco, which is for sourdough bread. And whatever, that's pretty self-explanatory. Beer, we're going to ferment some carbohydrates. So we have yeast that ferments carbohydrate into ethanol and carbon dioxide. So this is the bubbles, carbon dioxide, and the ethanol, the drinking alcohol, ethyl alcohol. It's drinking alcohol. So alcoholic beverages vary in alcohol content and flavor depending on the starting materials. Of course, yeast, anything that's added in during it, and we could have other bacteria that could be used for fermentation as well. In addition to yeast, remember, yeast is a eukaryote; it's not bacteria, but it's still a microbe. But whatever. And then we have ranking of the bitterness of beer based on the IBU. I don't know, I don't care about this, but hey, here it is if you're interested about it. Basically, India Pale Ales. I don't drink IPAs because they are so bitter. I don't care for them personally. So this is essentially how beer is made. You have the malting of the barley, creating of the mash, the wort with the hops, and then fermentation, and then your beer. I don't know what it means. And there's more slides associated with it. And if you guys are interested in it, I do encourage you to look it up. But, and there is some on the lesson that is associated with this chapter, but I don't know anything about this. So I'm not going to test you over it either, but if you're interested, please feel free to look it up. Wine, we have fermentation of grape juice, have the creation of must, which is the juice given off by crushed fruit. And that serves as a substrate for fermentation. Yes, the bacteria from your feet can have an effect on it. Bacteria that, or the yeast and whatever, the bloom is what they call it on the outside of the grapes, the biofilm, the stuff that grows on the outside of it. You know, you've seen grapes before where like the dark ones that have like that film almost on the outside. That is what I'm talking about. That's the bloom. So that's anything, any bacteria on the outside that could have grown on it that has to do with the taste of it. So anyways, get the must, you crush it, you get the must, that's the liquid. White wine, you're going to have white skin grapes, and red wine, red skin grapes, duh. And you can mix them. So if you're going to properly ferment your wine, it should have 12 to 25% glucose or fructose in it in order to get the appropriate amount of ethanol by volume. 7 to 15% is what you want. You can get more if you add other things into it, but it's whatever. I'm not going to ask you about that. So yeah. So you use wild yeasts that are on the outside of grapes. That's why they talk about like location a lot when they're talking about wineries and stuff, because you want it to have a good location where it's favorable to certain kinds of yeast. So you can get that good bloom on your grapes. What else is important with fermentation? We have sauerkraut, you know, fermenting cabbage, essentially. If you've had kimchi, you know, I don't like these things. I like sauerkraut, I'm not gonna lie. I like a pickle too. Salt pickles and whatever. These are the bacteria or the yeasts or whatever is involved. Usually on this end, it's typically bacteria like lactobacillus that is going to be causing that kind of souring of that whatever you're eating, pickle or whatever. Then we can produce vinegar, which is typically when alcohol that we made in your wine or other plant juices, when that alcohol is oxidized to acetic acid. Acetic acid is vinegar, literally the same thing. And then that'll also have other things in it as well. If we don't filter it, and you can get it super pure, but we're not going to get into that. Yeah. Okay. And milk, now we're getting into cheese, right? That's where cheese comes from, is milk. Duh. Anyways. So we have curds, which is just coagulated milk, right? You guys know about curds and whey. Curd, if it's curdled milk, it's kind of got solidified process going on. You're going to talk about this a little bit when we get into the lab about the physiology of bacteria. So I do want you guys to be aware of the term there, but anyways, we can coagulate the milk with the addition of this rennin, which is a casein coagulase. So that particular protein in milk, this causes it to coagulate. Rennin does that. And then whey is the watery part. So we can separate out the solid part and make cheese from that. And you can treat it a lot of different ways in order to get certain kinds of cheeses and treat it accordingly. So this is just showing a guy laying out some curd. And we also have these milk products that we may or may not be familiar with, such as yogurt. You should be familiar with that, but it has lactobacillus in it. Lactobacillus gives that nice tangy, tart taste to it. And we have kefir, kummis, buttermilk is buttermilk. People don't realize, but it is a little bit fermented due to butter making. And then kombucha. Those are all fermented products. In World War II, yeasts and molds were added to food to supplement the diets of undernourished citizens because it was easy to grow up these microbes. And then we have several countries that mass produce yeast, bacteria, and algae as food intentionally. And we have something called protein. That is this. And then mycoprotein. And this is this. It's a mold. And then spirulina, which is a cyanobacterium. So a lot of us may have heard of spirulina. That is a cyanobacterium. It's a photosynthetic bacterium that we use as an alternate to green plants, and supposedly has all this, beneficial qualities in it. So getting into the foodborne diseases portion of it. 48 million people a year suffer from some sort of food infection. We have the Food Safety Modernization Act that changed the focus of FDA from prevention of food outbreaks as opposed to prevention of food outbreaks, as opposed to what they were doing before, was just investigating after the fact and then going back and trying to see why it happened. So now we're trying to prevent it instead. So thanks to that. So that's good. Everything around us that we can do around the food that can affect it include the following: care in harvesting and preparation. That includes washing your hands before handling. You don't want to go to, number 2 in the bathroom and you didn't wash your hands and then come out and handle some food and now you've got E. coli all over it. That's gross. So that's an idea there. We have destruction or removal of microbes. Just make sure in case somebody did get some stuff on it, or it didn't get cleaned well or whatever. Things like pasteurization. I'm sure you guys have heard of that. Cooking is a way to kill microbes. Make sure everything's cooked through. And then filtration we can do with liquids. And then we have prevention of growth overall, is why we keep things in cold or freezing temperatures. We can also preserve things in salts and stuff. So preventing food spoilage. If your body temperature is here, this is where a lot of the pathogens are going to be the happiest. So you can inhibit or even destroy bacteria at the below 0 point. So this is freezing, so it's a little bit above it. I'd say 4 degrees is great for refrigeration. And then if you want to freeze, it can even kill some things. So anyways, the point is then you want to get microbes up into this area at least, in order to start killing them off and making it safe to eat. We can also preserve things, not just with cold or by cooking. We can use organic acids to preserve things, nitrogen salts to preserve, like cured meats, sulfur compounds, oxides, and salts and sugars, literally. So if you have like a jam that's a lot of sugar in it, like people will make jams or like preserves, strawberry preserves or whatever, there's a lot of sugar in that. So it has a high osmotic pressure when we talked about things going in and out and osmosis and all that. So that all that sugar on the outside will cause the microbes to die because they're going to get sucked dry by their environment. So we also have desiccation, which is just drying the thing out, but it doesn't always kill the microbes. And bacteriophages that can be specific to target certain bacteria to kill them. In industry, we can also use bacteria in bulk to produce other compounds in bulk, such as antibiotics, hormones, vitamins, acids, solvents, and enzymes, which we kind of talked about previously, but we can do it on a large scale is what I'm trying to say. When we're talking about industrial microbiology, that's really talking about large scale. We're not just we discovered it and a few people are getting this. We're talking about mass production. So just to summarize this chapter 25, we talked about water treatment. We talked about products that can be made from fermentation. And we talked about the safety of food. So that's it for chapter 25. I hope that was super interesting for you guys. And then I will see you in unit 3.